it changes a regular codon into a stop codon. So a codon is basically a sequence of three nucleotides in DNA or mRNA. We haven't really gone over mRNA yet, but all you have to know is it's a sequence of three nucleotides in DNA. Um, and when we have a stop codon, it's basically a codon, but it signals for the protein synthesis in the cell to stop. So if there's a change in um, if a regular codon changes into a stop codon, then that can make the protein shorter than it actually should be, which can make it non-functional because it will stop protein synthesis before it's actually ready. Does that make sense? Feel free to ask any questions if you don't understand that. So then we have a silent mutations. So silent mutations basically alter a base, but it doesn't change the amino acid because of the redundancy of the genetic code. So that can be confusing, but just take notes of that and you can look into it. Or ask me any questions if you have any problems with that. So yeah, these are all types of point mutations. They're basically where only one base is substituted and changed in a sequence. Okay, great. So now we have insertions. So insertions are basically mutations where extra DNA bases are added. So let's go over the impact of insertion. So basically shifts the reading frame of the gene during translation, leading to an entirely different amino acid, and usually makes the protein non-functional. And I know I just went over some terms that you might not be familiar with. But yeah, we'll most likely be going over some of these terms later on. But all I have to know is that it changes the protein and it might make it non-functional, so it won't be able to carry out the job that it's supposed to do. Okay, then we have deletions. So it's basically the opposite of insertions, where we have mutations um, that make DNA lose or remove bases. So it's basically where bases are missing. Eugene asked, how common is it? In general, mutations are pretty common. A lot of people have mutations in their DNA. It really depends though, because some mutations can develop over time, and they affect genetic disorders that we'll be going over later in this presentation. So it really depends on like your environment, um, and also your genes. So yeah, getting mutations is pretty common, and um, yeah, because the genetic disorders are going to be going over today are all caused by genes. So yeah, don't worry, we'll go over that in a little bit. So impact of deletions. So like insertions, they cause a shift in the reading frame, which can make a non-functional protein. So these are kind of examples of negative mutations. So it can like harm the protein if it's executed improperly. So here we have frame shift mutations. So frame shift mutations happen due to insertions or deletions, disrupting the reading frame of the gene. So basically, a frame shift mutation is the same thing as insertion or deletions, but it's just a way to categorize both of those. So frame shift mutation is either insertion or deletion. So it's the same thing. Just another term to know. So yeah, as I already talked about, the changes in the sequence can make like a non-functional or malfunctioning protein. So here are some examples. Sorry. We have first up is point mutation. You can see in the top left corner, we can see that the difference between the two sequences, between the original sequence and the point mutation, is that one of the bases which changed. So instead of T, we have C. So that's an example of a substitution where there's a mutation in the sequence because one of the bases is different. And then to the right, we have an example of deletion. So you see how the T was taken out on the bottom where deletion occurred? So basically what happens after it's taken out is that all of the DNA is going to shift. So that's a frame shift. See where the arrow is going? It's all going to shift to the left in this case. So that's what has, so it can have a big impact on um the DNA because everything is shifting by one if a base is deleted. And then same thing with insertion, except it's one being added. So you can see there's a frame shift, so everything is moving to make room for that added protein. So the C wasn't there before, but it was added through mutation. Does that make sense? Any questions? Okay, so also another mutation type is duplication. It's kind of like insertion, except it's basically involving the repetition of a certain segment of chromosomes, which leads to an extra copy of the genetic material. So it's just like duplicating um, a certain segment of DNA. So for the impact, that can result in the production of extra proteins, which can disrupt normal functions of the cell. Um, but duplications can also provide genetic variation, which can be beneficial in evolution. So yeah, we'll go over later on how these mutations can actually affect us in the real world. So we also have inversions, which you might confused with which might be confused with insertions, but it's important to know the difference. So basically, inversions occur when a segment of the chromosome breaks off and it's kind of reinserted in reverse. So I'll show you what that looks like after we go over the next type of mutation. So yeah, this can lead to altered gene expression patterns, and it can also cause genetic disorders or diseases. And we also have translocation, which can be kind of difficult to understand, but it's basically where we move a segment of one chromosome to another, either within the same chromosome or to a different one. So that can result in gene disruption. It can alter gene expression or fusion genes, which can actually contribute to cancer development. So here is the diagram. It might be a little bit blurry for you, so sorry about that. But we can see in the first box where we have duplication, you see that there are two B's when there was only one before. So that's an example of duplication. See, B was duplicated. Then we have inversion. So you can basically look at the difference between the before and after. So see how B and C were on the top and now they're on the bottom. In kind of like a reverse order. Yeah. Then we have translocation. So just take a moment to try to see what's different between the before and the after. So we have two different chromosomes. Basically, what happened was that some of the bases got switched off. So you see the bottom one, it got switched to the other chromosome. And yeah, some of the bases kind of like switched between chromosomes. So yeah, that's an example of translocation. And if you don't understand that, feel free to ask me, or you can look at diagrams online that might help you understand it better if this is too blurry or you might not understand it now. Okay, so why is it important to know about these mutations, like all these different types of mutations? And there are actually a lot of other different types of mutations, but these are the basic ones. And I could go into way more depth on this, but obviously we don't have time for that since this course is pretty short. But basically, it's really important to understand these types of genetic mutations so we can actually understand how complex genetic disorders are and like how they occur and how we inherit them. So different mutations can have really different impacts on our health and development. So yeah, we'll be checking out some of these genetic disorders pretty soon. So first, let's go over some of the causes of mutations. So first, there are spontaneous mutations, and these are basically naturally occurring mutations due to errors in DNA replication. So these just happen when your DNA is replicating, and there's just an error in replication, so you naturally get a mutation. So that's not really something you can prevent necessarily. Meanwhile, there are induced mutations, and these are basically caused by external factors. That can be radiation, chemicals, or certain biological agents. I'll show you I'll show you that an example of that. So let's see. This is the structure of DNA. Remember, it's in a double helix. Um, so there's an incoming UV photon. That's what the arrow represents. And if you don't know what that means, it's basically kind of like a ray from the sun. Sun gives off UV rays. So we have this incoming UV photon and it's headed for the DNA. So let's see what happens afterwards. In the after picture, you can see that the DNA totally broke and now there's like a mutation there, right? So that's just an example of how external factors can cause mutations. So now the structure of DNA was changed. So yeah, that's an example of external factors. Any questions so far? Okay, let's move on. So here are the effects of mutations. And like what kind of effects they can have at least. So first we have neutral mutations. So these are basically mutations that just have no effect on the organism. So they don't really do anything. They're not harmful, they're not beneficial. They're just mutations that don't have any effect. Then we have beneficial mutations. So these basically enhance the organism's traits and improve its survival. So that can be like natural selection. That's how we evolve through beneficial mutations. Got it? So then we have harmful mutations. So most of the time, we'll be studying harmful mutations outside of natural selection for beneficial mutations because harmful mutations are what cause genetic disorders and they lead to health problems. So now we're going to go into some common genetic disorders. I know I got some questions about these during one of my previous lessons, so now we're going to have the chance to go over them. Okay, so the ones we're going to be going over are cystic fibrosis, Down syndrome, sickle cell anemia, um, and yeah, you can ignore the last thing. We're going to be going over another one. So basically, genetic disorders are conditions caused by changes in the DNA sequence that are obviously harmful to us. So first, we have cystic fibrosis. So basically, this is a genetic disorder that affects the lungs and digestive system. It's caused by mutations in the CFTR gene, and it disrupts the flow of salt and water in and out of cells. So this leads to thick, sticky mucus, which makes it hard to breathe and digest things. So oh, sorry, I don't know what happened with that. I think it just refreshed. Going back to slideshow mode. Okay, so the CFTR gene mutation. Let's go over that in a little more depth. So basically, the CFTR gene provides instructions for making a protein that regulates the movement of salt and water in and out of cells. So that's the protein that controls that, right? Um, so this gene is really important for like breathing and digestion. So when there's a mutation in that gene, it basically disrupts its function. So it can make it, not function correctly. And when it's not functioning, then the flow of salt and water in and out of cells is going to be also disrupted. So that leads to cystic fibrosis. So some of the symptoms are chronic cough, lung infections, difficulty breathing, digestive problems, and then some treatments include medications, airway cleaning techniques, and lung therapies. Any questions on that? Or like about the CFTR gene mutation and how that works? Okay, let's move on to Down syndrome. This is another example of a genetic disorder. It's caused by the presence of an extra copy of chromosome 21. So this addition can lead to developmental and intellectual problems. So yeah, people with Down syndrome usually have like unique physical features and cognitive impairment. Like some characteristics and challenges of this include slower physical and mental development, characteristic features like I already said, um, heart defects in some cases, learning disabilities, speech delays, increased susceptibility to certain health conditions. So yeah, these are just case studies to show you guys like what kind of things can a genetic disorder look like. So next is sickle cell anemia. So basically, sickle cell anemia is a genetic blood disorder where red blood cells become misshapen and they break down. So that can lead to anemia, pain, and a lot of other complications. And it's caused by mutation in the HBB gene, which alters hemoglobin structure. So the HBB gene, we can infer that this like regulates hemoglobin structure. So when that gene gets a mutation, it's not able to function correctly. So that leads to this genetic disorder. And Jaden asked, can Down syndrome be severe? Yeah, it can. It depends on how our body reacts and what kind of mutation we have. But yeah, it really depends. For some people, it might be different. Like you might, some people are more cognitively um impaired, and their development might be um, impacted a lot more than other people. So yeah, it can be severe. But most of the time, it's not too impactful on well, yeah, it is impactful on a lot of different aspects of life because it can cause like learning disabilities, and yeah, the development is really important for humans. So yeah, it can be severe. Anyway, back to sickle cell anemia. Some symptoms are fatigue, pain episodes, jaundice, delayed growth, treatments, pain management, blood transfusions, medication, to prevent complications. Case study. Huntington's disease. So this is the last genetic disorder we're going to be going over. It's a sorry, neurodegenerative disorder caused by mutation in the HTT gene. So it leads to motor dysfunction, cognitive decline, a psychiatric symptom, and psychiatric symptoms. So basically, this HTT gene, like we talked about with the other genetic disorders, mutation in this gene caused this disease because certain functions weren't able to work because of the mutation. Yeah, so symptoms for this one usually appear in mid-adulthood. The symptoms are like involuntary movements, emotional disturbances, cognitive decline, and difficulty swallowing. Treatments include supportive care, medications to manage symptoms. So as you've noticed, a lot of these genetic disorders don't really have like specific cures. It's meaning like medication and like support to manage um and deal with living these diseases. So um yeah, most of the time there aren't like straight up cures that can can help these diseases go away. Like with Parkinson's disease, for example, that's also, or why is it called Huntington's disease? I'm assuming it's probably like the last name of the person who discovered it. But you can research that uh if you'd like. I'm pretty sure that's why. Um, I've studied more about this disease, but uh, I don't specifically remember. Maybe I can do a quick search. Why is it called Huntington's disease? Yeah, so Huntington's disease is named after George Huntington, um, who described it among residents in Long Island. So yeah, it's basically like the last name of the person who discovered it, if you're curious. And also with Parkinson's disease, for example, there isn't a cure, which is similar to these other genetic disorders. And yeah, remember how we talked about how like environmental factors can also um influence mutations? Well, yeah, Parkinson's disease is an example of um of a of a disease where both environmental factors and just genetic mutations can impact the disease and cause it. So yeah, that relates to the next slide, environmental factors and mutations. So environmental factors like UV radiation and pollutants can induce mutations. These mutations might lead to genetic disorders or increase the risk of diseases like cancer. So it's really important to be aware of environmental mutagens. The mutagens are basically just the word for things that cause mutations, like UV rays. Got it? So yeah, for example, nutrition can pair with um, mutations to cause a certain disease. So a disease doesn't necessarily have to be, oh, it's just caused by genetic factors. Oh, this one's just caused by environmental factors. So it can be a combination of the two. Like I did um, a research report on Parkinson's disease, and that was a really important factor in Parkinson's disease, where both of these things were causes. So complex genetic disorders. So some disorders like diabetes and mental health conditions include a lot of factors involving both genetic and environmental factors. So this is exactly what I talked about with Parkinson's disease. So yeah, understanding this like interaction between the environment and genetics is really important for trying to find a cure, prevention, and like other treatment strategies. Okay, so now prevention and genetic testing. So genetic testing is a medical test that identifies changes in chromosomes, genes, or proteins. And there are actually some ethical considerations in genetic testing. So in the genetic world, there's a lot of questions about ethics, usually, about privacy, consent, and equitable access to genetic technologies. Um, so all of these ethical considerations, um, um, they're needed to guide like research and experiences all the time. So yeah, you can look more into that if you're interested. And then we also have genetic counseling. So basically, genetic counselors are really important for helping families understand their own genetics and the risks involved with their own genes to make informed decisions about their health. So yeah, support networks and organizations provide assistance um, for individuals facing genetic challenges. So let's talk about the future, like what's going on, what are we developing? We're gonna go a lot into this during our either next week's lesson or um the last week's lesson. So yeah, this is a lot. We're just gonna be going over it pretty briefly so you guys have an overview of what's happening in the genetics field right now. So we have CRISPR. I'm not sure if you've heard of it. you might have because it's a pretty popular thing that's going on right now. So it's basically technology that offers um a level of precision in gene editing that's never been reached before. So it's really exciting because it holds a lot of promise for treating genetic disorders. So yeah. Um, but also like we talked about with ethics, there are a lot of, ethical concerns about, consequences that we might not be able to see right now, and equitable access. So basically, this CRISPR technology, it's kind of called molecular scissors. It basically lets scientists precisely modify specific genes. So, it lets us target changes in the DNA sequence like mutations. Um, so we'll be able to treat genetic disorders a lot more easily. So yeah, in terms of treating genetic disorders, the ability to edit genes holds the promise of curing a wide array of genetic diseases from cystic fibrosis, which we talked about, to certain types of cancer. Researchers are exploring CRISPR-based therapies to develop personalized treatments to tailor to an individual's genetic makeup. So yeah, that has a lot to do with precision medicine, which is basically where, um, like personalized medicine, which is where um, we tailor specifically to a person's individual genes. So, we make sure that our treatment aligns with um, what a person's genes are. So that like, determine how they'll respond to the um like the treatment, for example. Okay, so now I'm gonna talk about a project that we're gonna be working on. But first, do you have any questions about the lecture and what we covered? Any questions? Also, I might want to add here, in terms of, personalized medicine, there's this field that's currently expanding a lot. It's called pharmacogenetics, which is basically where we develop drugs, but with and like treatments for certain diseases, but in consideration to people's specific genes. So, we test how certain genes will affect the outcome of specific treatments for people. So yeah, does that make sense? It's a really interesting expanding field. That might make us treat genetic disorders a lot more easily and open a lot of doors uh in the future. So yeah, that's just the cool thing I wanted to add. Sure, you guys don't have any questions about anything? Happy to try my best to answer any of them. Okay, then I guess I'll move on. So basically, in the coming weeks, I'm gonna be talking to you about a project, which it's not that big of a deal, but it's kind of what we're gonna be doing more long term because it's due when this course ends. Um, uh, or when our last lesson, uh, the day of our last lesson. So basically, for this project, I have all of the requirements and instructions on Google Classroom already. It's gonna be posted right after this class ends, but it's basically where I want you to choose um the concept that we've been learning about, uh, or any concept that we've learned about or covered during this course, and you can just do a more in-depth report on it. So that's basically just a bit. I just want to give you a heads up. Um, so yeah, and also a lot of you still haven't turned in some of your homework from last week or the week before that. So, or actually, I think most of you here have already done that. So I think it's mainly people who haven't been attending or haven't been able to attend the meetings. But I think most of you here have been doing a pretty good job of that. Bryce, did you have a question?
I accidentally turned in the wrong my last week's last week's homework. I accidentally turned in week's two's notes instead
Mhm.
of week three's. I just turned it in.
Okay, great. I'll give you full credit. Thank you so much, Bryce. Yeah, I remember I made a private comment for that. Yeah, great. Thank you. You guys, I think the people who are here, thanks for coming to the meetings. I think you guys have been doing a really good job on submitting homework. But if you haven't, just please try to get it in there because it's mainly for practice. I understand if it might be easy for you if you're like already if you already covered that stuff in your own schools. Just let me know like in a private comment or something. Um, if you're like, oh, I'm already like familiar with this material, so or anything. But yeah, I really encourage you to do that because I just make it for you guys to get some practice and to know that you guys really understand and are listening. So yeah, so this project, don't stress about it too much. It's just a way for you to learn about like whatever topic you want. And if it's a topic we haven't already covered, just let me know. And I'll let you know if you can write about it or not. So it's kind of like you're gonna be doing research. It's just a really good opportunity to explore whatever you want. Okay, so our fun fact today is that there are between 30,000 and 40,000 genes in the human genome. That's just a really cool fact. It just shows how many genes we have. So it's like our bodies are like super complicated. We have all these genes, and just imagine how hard it must be to, like catalog all these. Like scientists know about like most, if not all of these genes. Some of the functions of these genes actually aren't known super well. So just genetics is a really big field that's always expanding. Because we always have more to learn. There's always more to know about these interactions between proteins, genes, um, and how it affects our lives. So yeah, just genetics is a really cool field. There's so much to learn. So yeah, that's the end of our lecture. Um, so I wanted to ask you guys, what do you want to do first? We can either do, I'm not sure if we have time for both. Actually, we might have time for both. Would you guys rather do another quizzes, or watch a video on transcription and translation, which is a new concept that might be confusing for you, but I will be posting the video on Google Classroom either way if you don't get to finish it in time. It's only 6 minutes. So yeah, just put in the chat what you guys want. Do you guys want to watch the video or do quizzes first? And yeah, I'm fine with either one, so just let me know which one you wanna talk. So we got quizzes and then one for video. Does anyone want to be the tiebreaker? Or if you don't care, just write I don't care. Okay, well I don't want to waste too much time just to going over this. Let's just do the quizzes while it's fresh in our minds. And then we should be able to do the video. But if there isn't enough time to do the video, I'll post the rest of it on Google Classroom for you to watch. So yeah, I'm gonna stop sharing my screen real quick. And I'll share the quizzes. So give me a second for that. Okay, here you go, guys. This is how you can join the quizzes. Some of the terms are, or like some of the questions are based on things we've learned from previous lessons, so it's kind of a combination of of what we learned so far. Um, but yeah, it's more for just practice to see what you know. So yeah, join as soon as you can, so maybe we might have time to do the. Are any of you having trouble joining, or are you good? So, Jaden's joined, Bryce has joined. I don't know who chair is. Um, so either Nora or Sally still needs to join. I don't want to leave any of you out, so I'm gonna be starting it soon. But tell me if you want me to wait for you. Just you can just private chat me. So yeah, private chat me now if you want me to wait for you. Otherwise, we're gonna have to get started. Okay, I'm uh gonna be starting in a few seconds now. Okay, let's go. Good luck, guys. Hey, great job, guys. Nice job to chair whoever that is. Okay, looks like you guys did pretty good on this. Nice job. Okay, so we only have like barely enough time for the video. Oh, Nora said she shared. Great job, Nora. Nice job. Great job, all of you. You guys did really good. Seemed close. Okay, so we like barely have enough time for the video. I can go ahead and do it. I can go ahead and try to start it as soon as I can. So give me a second to pull it up. Hopefully it won't be too confusing for you guys. I'm hoping it's but sorry, that's an ad. Okay. So let me see if I can share my screen and audio. Share sound. I'm hoping you guys will be able to hear my hear my video too. So let me know in the chat if you guys can hear this.
Hey, it's Professor Dave. Let's talk about DNA transcription.
Can you guys hear that? Just shout yes or maybe put in the chat. Yes. Yes, okay, great. Thanks, Bryce.
And translation.
He knows a lot about the science stuff. Professor Dave explains.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish or a cat or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule, consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism. Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always, feel
Okay, guys, I hope that was understandable for you. If not, you can go back and watch the video again, maybe um research some of those terms you might not know. So that might be a lot for learning transcription and translation just from one video. But yeah, if you're interested in learning more about that, since it's more like about protein synthesis and not really as directly connected to genetics, compared to what we were talking about uh during this week. Yeah, so if you want to learn more about that, you can do some of your own research. But yeah, thanks for coming, guys. Make sure to look on Google Classroom for your homework. Bye, thank you. See you next